It's a pleasure to talk about a story that started in 2017 when we founded Landos to develop safer, more effective therapies for autoimmune disease, where there's a huge unmet clinical need. And the approach that we've taken is to look at the immune system as a massively and dynamically interacting system where targeting a single component of that immune system doesn't make sense given the compensatory mechanisms that are built in. So we've centered around identifying novel pathways in the interface of immunity and metabolism because we've demonstrated that those become great portals to impressive therapeutic efficacy, initially in Crohn's disease and ulcerative colitis, but also applicable to other autoimmune conditions. And we've accomplished that with a pioneering research platform based on advanced computational methods in the interface of AI, artificial intelligence, and advanced computational modeling methods. As a result of those efforts, we've identified and disclosed two top targets, the lanthionine synthetase like 2 pathway, which I'll talk in more detail in several slides during the course of this presentation. We've also disclosed a NOT-like receptor, a regulatory NOT-like receptor called NLRX1. But we have additional undisclosed targets also in the interface of immunity and metabolism that show significant promise in the development of transformative therapeutics for autoimmune disease. Our lead asset is BT11. It's an orally active, gut-restricted small molecule with limited systemic exposure that targets the lanthionine synthetase like 2 pathway. This asset is currently in phase 2 clinical testing for ulcerative colitis and will be advancing to phase 2 clinical testing for Crohn's disease. Landos is not a one-trick pony. I'll show you the pipeline later on. While we have BT11 as our top asset, we are about to advance another asset called NX13 to the clinic, and we have a pipeline of preclinical assets and some of them in IND-enabling stage. Landos, when it was founded in 2017, received a Series A financing round from Perceptive Advisors. And this round allowed us to move our lead asset from preclinical stage to phase 2 ready stage. And in August, we announced a Series B crossover round co-led by Perceptive Advisors and RTW. So we've made significant progress from founding the company, having a very early stage preclinical asset, BT11, in 10 million. We've been able to, in a cost-effective, lean and efficient manner, advance therapeutic for a new chemical entity, for a novel mechanism of action, where we did significant amount of work in terms of validation. We ran head-to-head studies of BT11 versus biologics, 5-ASA, JAK inhibitors, and we demonstrated that BT11 outperformed those. We did all IND-enabling work and phase 1 clinical testing in about 18 months. And as I mentioned, by August of 2019, we're ready to start phase 2 clinical testing, and we officially pulled the trigger on that phase 2 ulcerative colitis program after raising $60 million in Series B crossover financing. We have a leadership team with industry experience committed to the development of novel therapeutics for autoimmune diseases. We have people with experience in advanced computational modeling, translational and clinical immunology research. Some of them have been working together for the last 5 or 15 years. We have a strong finance expertise, clinical operations, and medical experience in phase 2, phase 3 clinical testing. Our board of directors is composed by our main investors, Perceptive Advisors, representative from Perceptive Advisors and RTW. And we have a clinical advisory board composed of the top key opinion leaders in the space of ulcerative colitis and Crohn's disease. Bill Sanborn at University of California, San Diego, Fabio Cominelli at Case Western, Francisco Silvestre at UNC Chapel Hill, Jean-Fred Colombel, Mount Sinai, Maria Breu, University of Miami, Florida, and Asher Kornbluth, also at Mount Sinai. So with this team, with this board, this leadership team, and this clinical advisory board, we've been able to advance some of the assets currently in the pipeline, BT11 currently in phase 2 clinical testing, NX13 in the later stages of IND enabling, which will advance to IND in Q2 of this year. And then we have other assets that target the LANCL2 pathway at the preclinical stage that have a different PK profile than BT11 and provide great opportunities for lupus, rheumatoid arthritis, MS, and type 1 diabetes. Over the next few months, we are going to continue to conduct the phase 2 clinical study in ulcerative colitis. We are going to advance NX13 to phase 1 human clinical trials. We are going to grow the company. The Series A was conducted in a very lean manner. We had 15 employees. Now we have 16 positions open that will allow us to prepare from perspective of operational, financial, and clinical readiness. And we're going to be advancing additional assets within the pipeline. The core of these discoveries is an advanced computational target discovery engine, which is based on machine learning, artificial intelligence-based analysis of gene expression and proteomic data from multiple sources, mainly translational data. And this platform allows us to identify blind spots or missing linkages in regulatory and immunometabolic pathways. That's how we contributed to the discovery of LANCL2, how we identified the power of NLRX1, and how we continue to identify novel therapeutic targets in the interface of immunity and metabolism. LANCL2 is a success story. It's a novel portal that allows us to modulate inflammation within the GI tract. We have, in the context of Crohn's disease and ulcerative colitis, significant immune cell infiltration within the gastrointestinal tract, production of inflammatory mediators. And what BT11 does is to activate a dual mechanism of action, which results in suppression of inflammatory mediators, those mediators that are well connected to the pathogenesis of Crohn's disease and ulcerative colitis, and at the same time, and that's very important and I think unique for drugs for ulcerative colitis and Crohn's disease, enhances regulatory T-cell responses. And regulatory T-cells are the anti-inflammatory arm of the immune system. If we can harness their anti-inflammatory potential, we have an opportunity to bring that inflamed intestine back into homeostasis. And we've described this in a simplified way in this slide, where we have our lead asset, BT11, acting and binding to LANCL2. BT11 is an agonist of the LANCL2 pathway. And as a result of this binding, it decreases those mediators of inflammation, such as TNF-alpha, interferon gamma, MCP1, IL-6, and IL-8. And importantly, it also increases anti-inflammatory molecules in regulatory T cells that protect from autoimmunity, from interleukin-10 to FOXP3, a master regulator in regulatory T cells, to immune checkpoint inhibitors such as PD-1. And as a result, BT11 helps generate a suppressive regulatory CD4 T cell phenotype that restores and maintains immune tolerance within the gastrointestinal tract. And we've published these results and we've validated these in a series of peer-reviewed publications. Now, the LANCL2 pathway is more complex than a multimodal mechanism resulting in suppressed inflammatory mediators and increased regulatory responses. As I mentioned earlier, Landos is focusing on immunometabolism. And we believe that those targets that are capable of not only modulating inflammatory and immunological pathways, but also changing metabolism, provide a unique opportunity to reprogram the immune system from a dysregulated, excessive inflammatory state into a regulatory anti-inflammatory state. So the LANCL2 pathway is a multi-pronged mechanism of action One of the effects of BT11 is to enhance the CD25, STAT5 signaling to support the stable differentiation of regulatory T cells, but it also increases pyruvate dehydrogenase activity, which results in increased oxidative metabolism supporting FOXP3 stability. There's no other drug in the market that currently accomplishes what BT11 can accomplish on the regulatory T cell side. And this has implications in Crohn's and ulcerative colitis, but this mechanism is also critical for other autoimmune indications. BT11 that regulates glycolytic pathways associated with TNF-alpha production and effector CD40 cells, including production of lactate and overexpression of NO1. And as a result of all of this, BT11 increases the suppressive effects of regulatory T cells due to enhanced immune checkpoint inhibitors. In terms of local activity, we've demonstrated that BT11 acts locally in the GI tract without systemic distribution. We can see that BT11 reaches the ileum, the colon. It's present in stools, but the amounts in plasma are very small. We've demonstrated in humans that the concentrations in the GI tract are 6,000 times higher than those in plasma, and the amounts in plasma are eliminated in a matter of 2 to 3 hours. So an oral dose of BT11, 99% of that will make it into the gastrointestinal tract, and about 10% of that will penetrate the intestinal mucosa, where it's going to engage the target, LANCL2, in regulatory T cells, in effector cells, and change that immunological microenvironment from an inflammatory state to an anti-inflammatory state, and therefore provide therapeutic efficacy. Is BT11 only going to be a mouse drug? We believe not, because we've run a significant amount of translational studies before even moving to the clinic in peripheral blood mononuclear cells, primary cells from humans. We've also run those studies in lamina propria mononuclear cells from Crohn's disease and ulcerative colitis patients. And we've asked the question, if we culture those primary human cells with increasing concentrations of BT11, do we see a change in those markers that we had observed in animal models to be modified, such as anti-inflammatory markers, IL-10 and FOXP3, or pro-inflammatory markers, TNF-alpha and interferon gamma? Do we see a change in a similar direction? And the answer is yes. Panels A and B show upregulation of IL-10 and FOXP3 in these human primary cells. Panel C and D show downregulation of inflammatory markers in those primary human cells. Moreover, if you look at panels E and F, you'll notice that when we disrupt the expression of LANCL2 by using siRNA, the ability of the drug to exert those immunomodulating abilities is abrogated, which further supports the selectivity of BT11 for LANCL2. We've run extensive studies in mouse models of inflammatory bowel disease. We've tested BT11 in 5 mouse models of inflammatory bowel disease. This data is from dextran sodium sulfate model, but we have 4 additional models. In 2 of these models, we've run head-to-head comparisons of BT11 versus other drugs that are currently used in the treatment paradigm of Crohn's disease or ulcerative colitis, including anti-TNF-alpha and 5-ASA. And what this data shows is that BT11 outperforms in this preclinical model TNF-alpha blockers and 5-ASA. That's shown in the leukocytic infiltration slide in the bottom of the slide. And if you repeat this study in mice that lack LANCL2, if we engineer mice that don't have the target, the ability of BT11 to elicit that therapeutic activity is abrogated. These effects on disease activity and pathology are correlated with favorable effects on biomarkers connected to response to treatment, such as fecal calprotectin. BT11 is more effective than TNF-alpha blockers at decreasing fecal calprotectin, at decreasing Th1 responses, which are connected to the pathogenesis of Crohn's, and you see increasing regulatory T cell responses and decreasing Th17 responses. So based on this extensive preclinical package, we decided to move to phase 1 clinical testing. This study was completed about a year ago. It's a SAD-MAD study where we tested doses ranging from about 8 milligrams per kilo to 100 milligrams per kilo in 5 cohorts of single ascending dose, 3 cohorts of multiple ascending dose, in about 70 subjects. And we asked the question, are there differences in adverse event profile even at the highest dose? And the highest dose is equivalent to 7 grams of BT11. It's a huge dose of BT11. Those subjects were taking about 17 tablets of BT11. So that was the higher cohort. The adverse event profile at any of the cohorts was no different than placebo. And we also wanted to ask the question, given that other drugs in the space cause a drop in white blood cell counts, will BT11 cause a drop in white blood cell counts even at those huge doses that we tested for safety? And this data shows that BT11 doesn't cause a drop in white blood cell counts, which suggests that the potential for BT11 to induce immunosuppression is limited. We were also able to demonstrate that BT11 is observed in high concentrations in feces, both in single and 7-day dosing, and there was a dose response. And we demonstrated that GI concentrations of BT11 are 6,000 times higher than the Cmax levels found in plasma, which validated the results of the animal testing showing that BT11 is actually a gut-restricted drug. So based on these results, we felt very comfortable testing about 500 milligrams and 1,000 milligrams in the phase 2. Moreover, we looked at fecal calprotectin levels. Fecal calprotectin is a biochemical measurement of the protein calprotectin in stool. It's considered an inflammatory protein. It's produced by neutrophils. It's a way by which gastroenterologists distinguish IBS from Crohn's disease and ulcerative colitis patients. It's also a predictor of flare-ups in UC and Crohn's disease. And the elevated fecal calprotectin indicates the migration of neutrophils in the intestinal mucosa, which occurs during intestinal inflammation. Even though those were normal healthy volunteers, and therefore the starting amounts of fecal calprotectin were very low, we're able to see a signal whereby all subjects treated with BT11 showed a further decrease in fecal calprotectin concentration. So that was very encouraging and was consistent with the results of our preclinical testing. If you think about the current IBD treatment paradigm, patients usually start from mild to severe. So starting from left to right, we have aminosalicylates, and they tend to work in about 40% of patients, sometimes 30% of patients, depending on statistics, and then they are moved to corticosteroids, immunosuppressants. Eventually, they move to TNF-alpha blockers, alpha-4 beta-7 blockers. The way we are thinking about BT11 and the way we are designing the phase 2 program is that in our ulcerative colitis trial, we are going to go after a population of mild to moderate patients. So basically, it covers the left side of this slide, after 5-ASA and into TNF-alpha blockers. Our Crohn's disease trial is going to go after a population of moderate to severe patients. So after immunosuppressants into the biologics. So it provides a unique opportunity for BT11 to tackle a multi-billion-dollar market. This is global data showing the market in 2019 now and how it is predicted to evolve in Crohn's disease and ulcerative colitis by 2024, which will be the first or second year in which we'll be commercializing BT11. As you can see, there is a significant dominance of TNF-alpha blockers, but global data is predicting that that market will be disrupted and will be other treatment modalities. We believe that there's an opportunity for a multi-billion-dollar market for BT11 in the ulcerative colitis as well as in the Crohn's disease space. Moving to the clinical testing that is currently going on. In August, we dosed the first subject, the first patient with ulcerative colitis. The design of the study that is going on right now is a double-blind randomized study with 195 ulcerative colitis patients with mild to moderate disease. Those patients are randomized into 3 groups: placebo, low-dose BT11, high-dose BT11. And we have a 12-week induction followed by a 30-week maintenance. In addition, we have an opportunity for the non-responders to join an open-label extension study. By the summer of this year, by the summer of 2020, we are anticipating to have the top-line results for the induction phase of this trial. We'll also be starting Crohn's disease study with two groups, the high-dose BT11 with 1,000 milligrams and a placebo. We'll also have a design of 12-week induction followed by a 30-week maintenance, and the non-responders will also be able to join an open-label extension. So the results of this phase 2 program are going to be coming up soon, and that will shape the next stages of business development for Landos. As I mentioned earlier, Landos is not a one-trick pony. We have a pipeline, and today I'll talk briefly about another asset that we feel is very promising in our pipeline and in fact is about to move to the clinic, NX13. NX13 is targeting the NLRX1 pathway, which is a pathway that when activated results in decreased reactive oxygen species and antagonism of nuclear factor kappa B. Nuclear factor kappa B is involved in the transcriptional regulation of inflammatory mediators such as TNF-alpha, IL-6, and other inflammatory cytokines that are also connected to the pathogenesis of IBD. We have significant preclinical data in 3 mouse models of IBD. We have data in human primary cells from Crohn's disease and ulcerative colitis patients, and the profile of NX13 is again an orally active, gut-restricted small molecule for Crohn's disease and ulcerative colitis. We're anticipating to be able to initiate a phase 1 study in Q2 of 2020, and this will be a SAD-MAD study similar to what I described for BT11 a few minutes ago. The first subject will be dosed by Q2 of 2020. In terms of the mechanism by which NX13 provides therapeutic activity, NLRX1 is a mitochondria-associated receptor with the ability to modulate immune responses, particularly in CD4-positive T cells. NX13, our drug that targets this pathway, has site-specific binding to NLRX1, and we've demonstrated that NX13 increases oxidative phosphorylation in immune cells, resulting in decreased nuclear factor kappa B activity and lower production of inflammatory cytokines. We have shown potent effects in IBD models, and we have some data in this slide where we compare NX13 versus TNF-alpha blockers, tofacitinib, and mesalamine. I would like to call your attention to the fecal calprotectin data, where NX13 is capable of reducing fecal calprotectin concentrations about 86% when compared to TNF-alpha blockers that decrease fecal calprotectin concentrations by 32%, tofacitinib by 26%, or mesalamine, which basically has no effect in this mouse model. In addition, NX13 was shown more capable of decreasing Th1 responses and leukocytic infiltration than the other drugs tested in this mouse model. So in summary, we believe that Landos is uniquely positioned to develop oral first-in-class therapeutics for autoimmune diseases. We've made significant progress already with our LANCL2 program in a lean, agile, and efficient manner. We've shown that we can use capital efficiently. Landos is about novel mechanisms of action. LANCL2 is an extremely novel pathway in the interface of immunity and metabolism. NLRX1 is very novel. And the other pathways that we have not yet disclosed are very novel, but at the same time, very powerful and with huge potential to regulate, provide therapeutic activity. In all our drugs, we have shown extensive animal pharmacology, mechanistic data, toxicology. We are focusing on drugs that have a benign safety profile and human translational data. We have a committed leadership team with autoimmune disease and biopharma industry experience, and we are effectively executing our phase 2 program with the idea of having that data for the ulcerative colitis study in the summer of 2020. We have a strong financial position with backing of investors co-led by Perceptive Advisor and RTW investors and a robust crossover syndicate. And we have several members of our team here. If you have any questions, we'll be glad to entertain those questions at the hallway. Thank you so much for learning about Landos Biopharma.